Imagine a future where humans are immune to all diseases, live longer, and even have enhanced cognitive abilities. This might sound like science fiction, but with the advancements in genetic engineering, it is becoming a plausible reality. Our journey begins thousands of years ago when humans first started manipulating life through selective breeding. They didn't understand the science behind it, but they were, in essence, performing the earliest form of genetic engineering. Fast forward to the mid-20th century, the discovery of DNA was a game changer. It led to a deeper understanding of life's blueprint and opened up new possibilities to manipulate it. The 1960s and 70s marked the onset of more direct genetic modifications. Scientists began inducing mutations using radiation and inserting snippets of DNA into organisms, fundamentally changing their genetic makeup. The 1980s ushered in the era of commercial applications for genetic engineering. Scientists and companies began to see the potential for profit and societal benefit. This led to the creation of genetically modified organisms, or GMOs. One of the earliest examples was the flavor-saver tomato, introduced in 1994. This genetically modified food had a longer shelf life than its conventional counterparts, marking a significant milestone in the field. However, genetic engineering was far from perfect. It was a complex process, often taking years to achieve the desired results. It was also incredibly expensive, which limited its accessibility. These challenges posed significant barriers to the progress and wider acceptance of genetic engineering. Despite these early advancements, genetic engineering remained a complex, time-consuming, and expensive process until the introduction of a revolutionary technology, CRISPR. This groundbreaking technology, which we will explore in the next segment, has the potential to change the game entirely. It's making genetic engineering more accessible and affordable than ever before, bringing us one step closer to that future where we're immune to diseases, live longer, and possess enhanced abilities. Stay tuned as we delve into the world of CRISPR. CRISPR, a game changer in the field of genetic engineering, has made gene editing accessible to almost anyone with a lab. It's like the Swiss Army knife of biology, allowing researchers to slice, dice, and tweak genes at will. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. That's a mouthful, right? But the concept is simple. It's a system that bacteria use to defend themselves against viruses. When a virus invades a bacterium, the bacterium captures snippets of the virus's DNA, integrates it into its own DNA, and uses that information to recognize and destroy the virus if it attacks again. It's an ancient immune system, and scientists have repurposed it for modern genetic engineering. The system is precise, efficient, and applicable to various organisms. It's like a pair of molecular scissors that can be programmed to cut and paste DNA at specific locations. This precision allows scientists to edit genes in living cells, switch genes on and off, or target specific DNA sequences. The potential of CRISPR is enormous. Consider this. Scientists have used CRISPR to cut out the HIV virus from living cells. That's right, they have effectively deleted a deadly virus from a cell's genetic material. This showcases the tremendous potential for treating diseases and even cancer using this technology. But what makes CRISPR truly revolutionary is its programmability. Scientists can design a guide RNA sequence that matches the DNA sequence they want to edit. This guide RNA directs the CRISPR system to the right part of the genome, where an enzyme called Cas9 makes the cut. This allows for precise gene editing in a way that was once thought to be the stuff of science fiction. This precision and accessibility of CRISPR have opened up a whole new world of possibilities, especially in the medical field. With CRISPR, we stand on the brink of a new era in medicine, with the potential to cure genetic diseases and revolutionize cancer treatments. CRISPR's power lies in its precision. Imagine a spell checker that corrects typos in a document, but this document is the human genome, and the typos are genetic mutations. CRISPR can precisely target and correct single-letter DNA mutations, the minute changes that can cause devastating genetic diseases. From cystic fibrosis to sickle cell anemia, there are over 6,000 known genetic disorders, many of them without a cure. But with CRISPR, we could potentially change this bleak outlook. But it's not just about correcting what's wrong, it's also about enhancing what's right. CRISPR technology has opened up new frontiers in cancer research. Clinical trials for CRISPR-based cancer treatments have been approved, and scientists are hopeful. The idea is to supercharge the body's own immune cells, enabling them to hunt down and destroy cancer cells. If successful, CRISPR could turn the tide in our ongoing battle against cancer. But as with any powerful tool, the potential for misuse exists. The ability to edit the human genome raises significant ethical concerns. The specter of designer babies looms large, as does the possibility of irreversible changes to the human gene pool. What if we could edit embryos to ensure our children are taller, smarter, more athletic? What if we could eliminate not just diseases, but also perceived imperfections? And once we start down this path, how do we decide what constitutes an improvement? While these questions are not easy to answer, they are crucial to consider. Genetic engineering holds the promise of a healthier, longer life. But it also has the potential to fundamentally alter what it means to be human. It's an exciting time, but it's also a time for caution and thoughtful deliberation. While the medical possibilities are exciting, they also bring us into uncharted ethical territory, especially when it comes to enhancing human traits. The concept of designer babies is no longer confined to the realms of science fiction. CRISPR could potentially make it a reality. Imagine a future where parents could customize their offspring, selecting traits like immunity to diseases, heightened intelligence, or even an extended lifespan. This might sound like a plot from a dystopian novel, but the advent of CRISPR technology is gradually nudging us towards this reality. CRISPR, with its precision and programmability, could potentially allow us to design babies to be more resilient against diseases that have plagued humanity for centuries. Imagine a generation of children with natural immunity to HIV, cancer, or even the common flu. The health benefits would be immense, reducing suffering and potentially freeing up resources in healthcare systems around the world. But why stop at disease resistance? With our increasing understanding of the genetic basis of intelligence, we could potentially use CRISPR to enhance cognitive abilities. We might engineer a future generation of Einsteins and Mozarts, accelerating human progress in fields like science, technology, and the arts. And then there's the tantalizing prospect of extending human lifespan. Aging is in many ways a genetic process. If we can understand and modify the genes that control aging, we might be able to significantly extend healthy human lifespans. Imagine living 2, 3, or even 400 years, all the while maintaining the vitality of a 30-year-old. Beyond individual benefits, widespread use of genetic modification could lead to significant changes in the human gene pool over generations. This could have profound implications, potentially helping humanity adapt to new environments and challenges. For example, as we venture out into space, genetic modification could help us survive and thrive in alien environments. While these possibilities are thrilling, they also raise serious questions about the future of our species and the ethical implications of such advancements. Despite the potential of genetic engineering, it is not without its challenges and ethical dilemmas. The road to unlocking the full potential of our genes is far from smooth. The first hurdle? Accuracy. While CRISPR is precise, it's not always perfect. Sometimes it can make unintended cuts in the DNA, leading to unexpected mutations. This could potentially result in unforeseen health problems, complicating the pursuit of cures for genetic disorders. Another challenge is the unpredictability of gene editing. When we tweak one part of our DNA, we can't always predict how it will affect the rest of the genome. This is because genes often interact with each other in complex and subtle ways. Altering one gene might have a ripple effect, causing changes elsewhere that we didn't anticipate. But the challenges aren't only technical, they're also ethical. With the power to edit our genes comes the responsibility to use that power wisely. One major concern is the potential for societal discrimination based on genetic traits. Imagine a world where people could be judged not just by their actions, but by their genetic makeup. This could create a new form of inequality, one that is written into our very DNA. Another ethical concern is the misuse of genetic engineering by authoritarian regimes. The technology could be used to create a genetically superior ruling class, or to engineer soldiers with enhanced physical capabilities. Such dystopian scenarios may seem far-fetched, but they are not outside the realm of possibility. As we unlock the secrets of our DNA, we must also grapple with these ethical questions. We must strike a balance between the pursuit of scientific advancement and the preservation of our moral and ethical values. We must ensure that the benefits of genetic engineering are accessible to all, not just a privileged few. As we stand at the precipice of this new era of genetic engineering, it is crucial that we navigate its advancements responsibly, considering both its immense potential and the ethical implications.